Good morning, everybody, and welcome to the Science House Impact Zone. This morning, I'm very pleased to present to you the team for the Human Immunome Project. It's a global initiative to decode the human immune system using AI and building the first predictive models of human health. Today, we have the executive chair, Jay Metcalfe, who is also the legendary co-founder of Wired magazine, and is the visionary leader behind this cross-section of technology and biology and science, and drives the strategy behind the HIV. And Shai, who is our co-chief science officer, a world-renowned expert in systems immunology, and a professor at Technion, where he works on developing a scientific framework for translating massive biological data into medical breakthroughs, so making things happen on the ground. We'll do a 20-minute or so presentation and then have a chance to engage and ask questions and take us through the session. Jane, over to you.
Great, thank you, Emmanuel. So I'm going to just come over here and say a few words first about, Wired magazine was about the digital revolution, and we were very consciously starting a movement. We came to Davos. My first time in Davos was literally 1994, with the intention of telling the powers that be that the whole world and all the power structures were about to be upended. They didn't really listen, especially the media people, but it set us on a mission, and I think the impact, of course, of the digital revolution has obviously been felt for decades now. I came to the science field, and particularly, this life science research, via a couple of other things that happened. But the thing that I realized was the next stage of the digital revolution was when we turned our engineering mindset to engineering human biology, our own biology. And I was at a conference, and I was meeting all these people with 10 to 15 years, like Shai, of highly technical and specialized training, who were also hackers. And I realized, wait a minute, we used to say the digital revolutionaries were the most powerful people in the world. Now it's like the medical people and the researchers are the most powerful people in the world because they have the ability to transform our species in our lifetimes. And CRISPR had just been announced. We were starting to hack the brain. We were hacking food in really interesting ways. And I just thought, this is a new revolution. I called it a neo-biological revolution, new biology driven by digital technology. And so I started a new brand. It was called Neolife, now called Proto-Life, to cover all those changes at the forefront of genetics and neuroscience and longevity medicine, the future of food, et cetera. I got recruited into a precursor project to this. And part of what really set me off was a researcher at Yale, Carolyn Mazur, who was talking about women's health. And I thought, reproductive health, and what about non-reproductive health that relates to women? But the thing that really struck me was her telling me in 1993, there was legislation in the United States that said, if you get federal funding for your research, you must have female mice and female participants. And I thought, wow, in 1993, we have to pass a law to make that happen? And here's the crazy thing. 25 years later, in 2016, they passed another law that said, and if you don't, we'll fine you. So it's like being a woman, which is, of course, more than 50% of the population, was not considered baseline science. This was considered a little too complicated. Let's just see if we can make this work in male mice and in male humans. And I was so outraged by this and all of the follow-on, inequities that entailed. And then there's this whole idea that we're not going to publish the results because it didn't work in women, so we're just not going to tell anybody about that. That was another part of the legislation, is that you have to, publish negative results, which is still not really happening. The fact is, women are less likely to be diagnosed with heart attacks. They go into a hospital, present with heart attack symptoms, but because they're not as easily recognizable in a woman as in a man, they're oftentimes sent home. Cardiovascular medicines don't work as well in women. Women are more susceptible to autoimmune diseases, more susceptible to dementias, and so why are males, and particularly European males, considered baseline? Who deserves to be considered baseline, and who's been missing from this conversation? COVID was my first exposure to human immune response variability. I have a friend, most of us got COVID and kind of brushed it off. I have a friend who ended up upside down on a ventilator in a medically induced coma. You know, all of us have cancer cells, but in some people they start to become malignant, and in a small subset of those, they metastasize. You know, people die with brains full of plaques and tangles, but some people develop dementia. You know, what is it? Why are these changes persisting? So we have the tools now to start answering that question. We have the technology to measure and model systems immunology, taking the technology of systems thinking and bringing it into the medical sphere. And we can do this on an individual level as well as a population level. And so when we can tell you what your immune system state is on a deep level, and then make predictions about your future, health strengths and challenges, we'll have the most powerful tool currently imaginable for building truly precision-based medicine. So, I have the honor of working with Shai, a systems immunologist and amazing hacker, who is the co-chief science officer of this initiative to decode the human immunome. And I want him to talk about the science and the project, and then I'll come back later and talk about impact.
Thanks, Jane. So really a pleasure to tell you today about this amazing, ambitious project, but I'm going to take this to a very personal level. Imagine this is you asking the physician, hey doc, how's my immune system doing? Right? Sounds like a relatively simple question, right? In fact, it's not. The physician, and I suspect most physicians will agree with me on this, really has no clue on this. And if you're feeling like maybe there's a good answer, let's do this comparatively. If I were to ask, how's my heart doing? Then the physician has a lot of ways of monitoring it and changing your lifestyle or doing various interventions or treatments. That's not the case for the immune system. This is still mostly a no clue issue. Now the question to ask is, why is it that in the 21st century, when you ask a physician this question on a pretty important system in the body, this is the system that's in charge of basically maintaining homeostasis, keeping us safe, monitoring the environment all the time, right? It's basically a sixth sense, right? Its entire role is to monitor internal and external environment and respond to it, right? So that we are protected. Right? And that sixth sense basically that role defines that it needs to constantly adapt, constantly change. And the problem is it's a very complicated system. A lot of cells moving around in your body. There's not even one organ for this system. It's distributed. They change all the time. And because we all live in the real world and we each experience our environment differently, it's going to be different between us, right? And so the question one may ask is, not, this gives the explanation to why maybe the physician shouldn't be feeling too bad about himself or herself in this, but rather that we need to somehow get at this problem, and maybe this is the right time to do it. And I'll show you why it's the right time. Though the first thing is to show you some numbers, right? So this is thinking about healthcare. We know that it's broken, right? And here's one way of seeing it. This is the increased GDP in the U.S. of healthcare costs. Right, and it just keeps going up, and this is not just in the U.S., this is true in all countries, unfortunately. Sick care, which is really what healthcare is, right? We're dealing with disease, not with health, is currently unsustainable, and it's going to become even more unsustainable because the last period of life is where most costs go, 50% of the cost of healthcare, and we are an aging world, right? 65 plus is the new norm, right? Right now, 1 in 9 people in the globe are over the age of 65. In 20 years, 1 in 4. Okay, that's the numbers. It's already unsustainable, right? And we need to somehow deal with this, right? And the answer is obviously early prevention. But early prevention requires robust sensing, requires you to be able to monitor health and disease all the time. Remember, what's the system that's in charge of monitoring, right? And it requires to be able to do this around everywhere in the world. And this system is highly variable, right? There's population heterogeneity. So, the immune system is the sentinel. It's involved in most diseases, and its role dictates that there's variation. It's actually the largest, most highly variable system in the body, right? You can zoom in just into drug development, which is a well-known problem. The return on investment for internal R&D in pharma today is basically on the borderline of being negative, right? It sits at 8% and keeps dropping. The two peaks you see here are successive COVID vaccines and most recently GLP-1, but this is going down. You basically, R&D of drugs is becoming more and more and more difficult and just not profitable. And this is a barrier for a word we all like to hear, which is precision medicine. Because if I can't make money on it, right, how can I develop a drug to a smaller population in which I'll make less money on it, right? And so I can't actually go, there's a barrier here, right? And if you look at why this happens, the biggest failures that happen in clinical trials are currently in phase 2. This is where we test the efficacy of drugs, right? We don't understand the disease biology in humans. Mice are great. We've cured every disease. Humans, less so. No mice in the audience, I suspect, right? So we don't understand in humans, and we don't understand the variability, right? So even if we understand the disease, we go to Japan, and suddenly the same drug doesn't work, right? Example, malaria vaccines, 90% plus response rate in the US, less than 20% in Africa, okay? So this is the problem, and again, if you think about it, if I wanted to make one kind of change, one revolution, the way to address all of this for all diseases is actually by targeting the immune system, right? Solving this one disease at a time, one treatment at a time is too slow and costly, but if I understood baseline variation of the immune system, then I would actually be able to understand this disease biology much better because it's involved in every disease, and I'd be able to understand population heterogeneity because this is the system that deals with the response. And here's the good news. See here on the left, this is the complete blood count, right? The white blood cell count. People know 100% of your immune cells that you can measure from blood, and we've been doing this in humans since 1957, right? More automation and so forth, but basically measuring the same thing. The parentheses are still not adapted to the population variation. The last update was like in 94 and like 30,000 people in the US. And what you see on the right is a new technology. This is not even a new one. It's an old technology in the days where 2010, this is a single individual, all of their immune cells that you can measure in blood. Just much higher resolution, right? So if you look at lymphocytes here, right? I can do this. No? Yes. All of the ones here, I'll do this here. All of the ones here, these are all lymphocytes. Each has a different function, each has a different role, and they're just averaged here, right? And so all of immunology, all of basic immunology has been about figuring out what these do. And we can go into much higher resolution to this. Every point here is a particular cell type, but it's actually, even within that, higher resolution, tens and hundreds of different other cell types, right? Each of them has a function. We are not characterizing them currently today in modern day medicine. And we can do this now. So this is a modern day, if you like, CBC count for the 21st century. So if we want to actually answer that patient's question, how my immune system doing, what would we need to do? So we would need to deeply measure the immune system. We need to do this at the breadth of the population, of the global population, to understand the variability. We need to actually do this in baseline healthy individuals across many different places and understand really, because baseline is not very well defined. We need to challenge your immune system. We need to know what it looks like and what happens now when I perturb it, say with the vaccine. How do you respond? What does your state mean to how you will respond to a drug? And ultimately, because it's so variable, we need to track it over time. It's not enough to get at one time point. And therefore, we think it's the time to decode the immune system. The Human Immunome Project is basically about getting an understanding of baseline variability of the immune system, measuring this in hundreds of thousands of people, multiple time points, on the order of about 5 million samples is what we're trying to get. If you're looking at this, why do we think this is the right time? Well, the tools, as I just showed you one of them, the tools to measure the human immune system are here now. Back before 2010, this was science fiction. Now we can do this. We have tools for every piece of this. And if you like to think about the equation, the equivalent to the Human Genome Project, it took 9 years to develop the tools to sequence the genome. Project started in 1990, 9 years to develop the tools. We sequenced the last 4 years, right? The past 15 years, we've developed the tools to measure the human immune system, which we used to not be able to do. The AI is here. We know now how to make sense of this data, and we can show that it makes sense. And COVID made the public awareness and global leaders aware that we really need to actually get at baseline variability and understand the human immune system. And so this is the call for the human immune system. We're not doing this alone. So as Jane and I met in a conference 3.5 years ago with John Sang, my co-CSO. John is a systems immunology based in Yale, and he's also one of the leaders of the CZI Biohub in New York. Not with us today, but really my partner in crime in putting this plan together. And the vision of HIP and its mission is basically to develop a new future that harnesses the power of the immune system to improve health for all. And the mission to do this is to actually generate that immunological data set, the huge database of how the immune system looks like, and build this to be publicly available, to build models of the immunome and that are able to be predictive and accelerate medicine in all the different layers, if it's drug development, diagnostics, or others. Why now? So Jane already told you there's a sinking reality that the knowledge that we have about the immune system is lacking, right? COVID showed us this. We know about a huge amount about mice immunology. 80% of papers in immunology are published on mice. 80%. Hear this, frontiers people? Right? So we need to move to doing way more human stuff. We do some mostly in males and not females, despite the regulation, as it's still the case. It's still done in narrow set of populations around the world. And what we measure on those humans are mostly the clinical historical things that we've been measuring since 1950, rather than the new tools that we can actually get much more at. And those things we measured in 1950 weren't the right things. They were just the things that we could measure in 1950. And then they just stuck around. It's very hard to move something out of the electronic medical system. I've mentioned already the global population is aging around the globe. All of these problems are going to become even more acute than they are now. It's already unsustainable, and there's a huge transition around the world of urbanization that's causing what we call epidemiological transition. Infectious diseases are slowing down in many places. There's much more chronic diseases on the rise, even in LMICs. And so this is, we believe, is the right time to do this. Why does it need to be global? People often ask us this, right? Why can't I just do this in my country, in my population? Well, the problem is that if you actually want to understand the data, you actually need to have all the outgroups, everybody that's variable for you. You need to make the association, right, between what's happening to you and whatever it is that you've exposed, and what are those things that relate to. And the way you do this is actually by generating the data across the globe with all the variation that exists in it. This diversity is a must for understanding. It allows us to also build the global infrastructure that if there's a new pandemic or there's a desire to now understand Alzheimer, well, now we have centers around the world that can actually generate the data needed and not discover that the biomarker we just discovered for Alzheimer only works in the US, right? We need the scale. We need to be doing this in standardization. And ultimately, if we do this in the right way, it needs to be open, federated data sharing for this project. We need to close the gap on the current data, especially for underserved populations. There are focuses that we think you need to go through. So it's not just about measuring immunity at breadth. You also need to be making sure that you're going to reach particular goals. And we've set 4 of those. One is global health, so understanding this variation. I mentioned the malaria vaccine. Why are particular treatments working in one place in the world and not in others? So we can design treatments from the get-go that will work everywhere, or tailor them so that we don't fail in drugs because of population variation. Sex differences. How are the diseases different between males and female? Pretty much this is true for every disease. We say about autoimmunity, but even diseases that are not autoimmune, you see variation between females and males in different phenotypes. Aging, right? Ultimately, how we vary over time and between individuals and within ourselves over time is key to actually being able to monitor. Those parentheses we put on the CBC, they need to be tailored to ourself. Right? And so we need to understand this. And if you do those 3, you actually can tackle the last problem, which is what we call the epidemiological transition, this shift that's happening in from rural to urban societies, where you can go to Tanzania and suddenly people the place that never had diabetes or Crohn's has those diseases. And we can basically understand there not only what's happening to those individuals, but also how that relate to the origin of disease in other places in the world, like here in Davos. So with that kind of idea, let me give you a bit of a primer on why we think, again, this would work. Again, if we look at the immune system, the way I like to think about it is that it is a cellular system. The organ are ultimately hundreds of thousands, millions of different cells. Each of them has a different function. This is the atomic unit, the quanta at which we think about the immunology. Immunologists are people running around chasing characterization of cells, and what they discover is that there is a lot of cell types, as I just showed you. Each of them has a different function. They interact with one another, and so if you have one of them that doesn't work properly, we're going to see various types of phenotypes. And again, it's ultimately about getting an emergent response that we call immune protection, seeing the big picture, right? It's cells interacting with one another yields, right, something that's is not the function of any one cell type. It is we are protected from whatever disease it is, and you can see, for those sitting in the back, Mona Lisa from coffee cups. So immune health is an emergent phenomena. It directly relates to physiology because everything in disease sits on the relationship to the immune system. And so we can think about, it's not enough to just measure cells one cell at a time and so forth, as we've been doing for the past 20 years with the revolution of single cell transcriptomics, but it's actually about measuring the immune system in its entirety and link it to clinical medicine, right? Not forgetting that link. If we forget that link, we won't be able to bridge ultimately to drugs. And so it's a shift back from reductionism to thinking about physiology, and this is what the Human Immunome Project is all about. We need to be measuring not just cells and not just genome, but also cell functions, cell interactions, metabolomics, proteomics, and so forth. You need to be integrating across all these different layers of biology to be able to understand those emergent responses. And we already have some proof points that this is working, right? So with a lot of AI, my friends and I, as they say, have been really kind of showing that it brings value to measure the immune system. I'll show you a few examples here in these 4 different studies. The first here on the top left is from Mark Davis's lab at Stanford. This is a study of mono and dizygotic twins, looking at them at different ages. A study led by Peter Broden, who's currently in Karolinska. 210 people, monozygotic and dizygotic twins, and what you can see here is as they age, right? As they age, you get more and more things that are different between monozygotic and dizygotic twins, suggesting that even monozygotic twins, right, when they experience life, if they're identical twins, they start diverging, right? We become more and more different from one another just by experiencing life, implying that genetics is not enough, right? Another one, looking at this is from my partner in crime, John Sang. Gender-specific immune responses. A paper studied, this is about 70 people looking at individuals who had mild COVID, not hospitalized, and then a year after getting the influenza vaccination. How are they responding to an influenza vaccine? Well, if they're female, it doesn't matter if they got COVID or not. If they're male, they're actually more responsive if they had a COVID infection ahead, right? So your baseline is actually changing. What you experienced before, your life history changes how you respond to the next event. This is what the immune system is all about, always adapting. A study from Mihai Natia's lab at Radboud, in collaboration, long-standing collaboration with folks in Tanzania. Again, numbers you can see, 250 people and in urban environments and 70 people in rural making transitions. They basically gave them 2 types of diets. The type of diet that you eat here, traditional Kilimanjaro diet, the type of McDonald's that you eat in the urban environment. And in 2 weeks, you got increase in inflammation, right? And then they changed the diet again and gave them a lot of fermented banana beer, and you could see a drop in inflammation just a week after, right? These things are actually changing, and then you see the shift in diseases, infectious diseases here, chronic diseases in the urban environment. And last, from our own lab, looking at immune aging, we've been studying the longest running cohort of individuals, of aged individuals over time since 2007. We've been able to define an immune metric. You can kind of see here, we can take a sample from you, project it on this reference, and we're able to give you a score that describes your immune age. And we've been showing that this is predictive of all-cause mortality, cardiovascular disease, vaccination responses, and a variety of other things. So we have some first version of a metric of immune aging or immune health. So, cool 4 studies. You can think about them as a half glass full. Wow, amazing, cool stuff we published in Nature. Or you could say we have a problem. That's all you did, right? And why did you only do this on like 100, 200 people, right? And why do you know so much about people in Palo Alto, right? All of those questions are good questions. Therefore, this is what we want to change in the Human Immunome Project. We think that if this is the diversity of the immune system, this is what we've known today, right? And we need to change this. But the problem is that this is done right now in probably less than 50 labs around the world. It's really expensive, and we can do this in hundreds of people. What we need to do is build a project that will let us increase the variation in an iterative manner and do so at reduced cost. So we need some kind of an immune monitoring kit, right? The necessity to do this at scale, we believe, would be the mother of invention. We need standardized data generation. We need robustness and simplicity of the ability to measure this, because there's a huge amount of variation across the globe in technical capability and clean environments. We need to be able to scale this, and we need to do so in a local way that increases scientific capacity, not just in data generation, but also in insight, so that we can empower each one of the healthcare systems around the globe with this. The immune monitoring kit, as we think about it today, is taking all the different layers of the immune system, measuring them each one with the coolest technology we can today, right? And then studying that data and realizing how do we measure less things, but know just about the same information because of all the correlations in the data, and how do we do so cheaply and more standardized? This is what the immune monitoring kit development is all about, version 1, version 2, and so forth. To do so, we need a network of people around the globe that will help us to do this. And so we've assembled a scientific partnership network. This is really the leading institutes in the world that can profile the immune system. We basically have people on every continent in the world. We are ready to go with this. And in fact, just in November, we had a launch of the Scientific Partnership Network in Banbury Center in Cold Spring Harbor. You can see us all here. We're all excited because we have the plan and we now need to execute. We also need the funding for this, but this is the plan. We're also excited because we're standing on the shoulders of giants. This same house, this same building, in 1989 is where the Human Genome Project was started. You can see here the founders of the genome. This has really been a foundational project, right? It generated the reference genome, it generated the tools, the data infrastructure to do this, and it generated the mindset of open science and releasing of data, right, on which we're building. And so we really feel like we're standing on the shoulders of giants. It also had some issues. Well, one is ultimately the genome was sequenced in 2003. Right? When it started, we had 4 diseases, 4 drugs for which we had genetic information in their labels. When it finished 10 years after, we had 40 drugs that had it. 13 or 15 years later, you had 100 drugs that have a genetic label, and now it's about 800. Right? Ultimately, 20 years after, and we're still trying to apply this information into outcomes. So slow impact, and really, if you look at who was involved, not enough industry, not enough clinicians, not enough people, not enough payers, people that could take this early on and adopt so that we'd be ready to make the impact. It had the unexpected limited impact on explained variation. We thought genetics would explain everything. It explains less than 5% of disease. And it makes sense because disease is also about living life. And so we need a system that it changes with life history, and that's the immune system. All of epigenetics sits in the immune system. And ultimately, it had the unintended consequences of we sequenced people from the US, very few, and even afterwards we need to have a few variation. We still don't have the African genetic variants, which constitutes 50% of variation in the genome. So it increased global inequity because the gap between the haves and have not actually increased with this. So all of this is to say, I think we have the plan, we have the team to get it going. This is, we really think is a huge thing, and we believe that this is really where immunology needs to go. Big science that can change minds, can change how we do work, and take medicine ultimately to the next level. So to finish this off, I'll let Jane here tell you about what we think would be the impact.
Yeah, awesome. Thank you, Shai. I was telling my friends, what's on today? And I was like, well, wouldn't you like to have heard from the founders of the Human Genome Project what they were going to do and what the impact would be? This is orders of magnitude more complicated and orders of magnitude more impactful. And, being with John and Shai is just the experience of a lifetime of my entire career. And, having the opportunity to do something so impactful is enormous. So, having said that, Big science sometimes gets a bad rap in Europe. It's like, well, that's taking money away from other researchers and it's going into this big boondoggle and when are you ever going to deliver results, But there are a lot of really excellent examples. CERN, of course, the particle accelerator was the prior slide, is a really great example. They brought together countries with competing political ideologies and researchers who would normally compete with each other and IP issues that would normally be very sticky and managed to build something that enabled all researchers around the world to run their experiments and to advance the science. They, relied on grants, but they also have an earned income component of what they're doing, which is really important. The space race is a perfect example of something that, generated enormous amounts of progress for humanity and lots of cool technologies. We talked about the Human Genome Project. Those of you who were here, I think it was yesterday, to hear Henry Markham talk about the Open Brain Initiative, and now he's turning a lot of that intellectual property into, monetizable, commercializable technologies. And so we're going into this with, a very clear mindset. Step one is what are our deliverables? And so, we're going to build the largest data set of this advanced immunological data that's ever existed. And it will be diverse. As Shai says, we know a lot about people in Palo Alto. That's not good enough in 21st century science and medicine. And we're going to get not only their genomes, but their phenotypes. So we'll get their data, but we're also going to get their life stories and information. What are you eating and how much stress are you under and do you sleep well and so forth. A friend of mine said this and I love it. Genetics, we thought we were all different, right? And we were so busy othering people all the time. And, in Davos, we come together and we celebrate those differences. But nevertheless, the genome is 99.9% the same among all humans on the planet. What differentiates us is our immunome. And it's the result of, it's the history of everything that's ever happened to you in your life. And so, having this, capturing that diversity is really impactful. We'll have these samples. We can create a biobank. We can let future researchers and future technologies come back and sample all of these different populations again with new technologies. As we talked about, nobody gets as excited about standardization as I do. I think it's a big deal. The fact that people are doing this work in 50 different labs around the world right now, everyone has their own process. Everyone's got their own reagents. They've got their own way of noticing, like even collecting the data and saying, we use saline solution. There's 13 different ways to say saline solution, and if you say, just give me your data, and it says saline solution in a different way, it's like you can't build effective AI models if you put a bunch of garbage in, And so creating these standards is going to be hugely impactful and is a potential revenue stream for the project to have these tools and the platform that we can then license around the world, making the data openly available to all researchers, but then having some sustainability by licensing the tools in the platform. So being able to quantify, this is what my immune system looks like now. Challenge it, this is how my immune system functions when you give me a vaccine, for instance, and then predict over the course of time what your health looks like going forward, how you might respond to the next COVID variant, how you might respond to this vaccine versus that vaccine, et cetera. So quantifying and predicting, and then what I won't say, but all the immunologists involved in this project ultimately want is to uncover the underlying mechanisms of action of the immune system. And so that's kind of where you wave your hands and go AI. But our deliverables are the quantified and predictive models of the immune system. And then, he's talking about these parentheses, this is what's considered a reference range, but what's normal for somebody in Rwanda is not necessarily the same for someone in Palo Alto. And he's got this great example of research in Bangladesh, where a 3-year-old Bangladeshi child's immune signature was more similar to a 30-year-old in Palo Alto. So that kid had been exposed to more different things that 27 years of life were required in a Western, context to accumulate. So we'll have new reference ranges. What if we just said, well, this is how women respond. This is what your white blood cell count should look like if you're a woman versus a black man versus somebody else, These are hugely impactful. We also anticipate seeing early biomarkers of disease. We're going to recruit 200,000 healthy individuals around the world, but over the course of 5 years, some of them will get sick, some of them will get pregnant, some of them will die. we'll see those early indications of disease, which are enormously important for precursors and predictive abilities. We'll find new druggable targets, right? So if we see this is what's happening, how can we then identify that? And not just druggable targets generally, but also for different populations. So our long-term impact, yeah, we're going to transform immunology, right? We're going to go from mapping, like, well, this is the T cell receptor repertoire, and this is what's happening over here, to zooming out and getting a, three-dimensional, four-dimensional over time view of the immune system. We're going to galvanize the development of globally relevant and population-specific diagnostics, medicines, vaccines, immunotherapies. We will be doing a technology transfer and training all the people that we work with around the world so that they can do this, which will have the impact of spurring local economic development and ultimately lowering both national and global healthcare costs. So I was asked by Science House to say, what do we want? Why are we here in Davos? And the fact is, this is a big, hairy, audacious goal, and we need a lot of help from a lot of quarters. So we want to partner with industry. I think there's a role for industry to play, whether technology or pharma or biotech. We're looking for governmental support. We want to go into these countries, respect their data and health sovereignty, but at the same time, we recognize that if you share your data into these models, we're going to advance everything for all of humanity. But there are partners with NGOs, both global NGOs, but also local NGOs. Be able to recruit these populations is going to require community collaboration. We can't recruit them. We have to identify the people within each community who can do that recruiting, who can create the trust in who we are and what we're doing. The health metrics, what is the ROI for the Human Immunome Project? This is not any of our competence right now, and so this is also one of the things that we're looking for. And I think Europe in particular is really interested in and good at doing this. And then, ultimately we're building a movement. We want to transform the way people think about medicine. We want to accelerate the implementation of this way of thinking into our research, into our drug development, and into our medical education, and ultimately in clinical practice. We want to move into systems thinking, that we're not just looking at things in isolation. And in the end, this is why we're so thrilled to be here at Science House. This slogan, open science, for solving global problems, is really impactful, and that's really who we are and what we're trying to do. And this is going to benefit all of humanity.